100,000 DMPs, differentially methylated positions per strain. So this is actually quite a large number after only 30 generations. Now they do behave actually very different from DNA mutations. Not going to show this you here, but they are very unevenly distributed in the genomes because this is affected by presence of transposons, small RNAs. What's also very different from DNA mutations is the high frequency of recurrent epimutations. They are rare enough that you would expect very few cases where the same change has occurred in exactly the same position. We actually see about 30% of the epimutation happening at the same place. And quite a few of those are actually occurring in more than one strain. So this makes it already very different from regular DNA mutations. The only other thing that makes it very different is the number of apparent changes per generation when you take the total number of changes and divide it by the number of generations. So if we compare generation 31 to this early generation here, the total changes, and divide that by the number of generations, we calculate 900 differentially methylated positions per generation. However, when we contrast these two, which are 6 generations apart, we come up with this number here, 1,300 changes per generation. If we actually compare parents and descendants, it's 3,300 changes per generation. If we look at siblings, it's a very similar number. So this is really quite odd. There's much less than linear increase in number of epimutations. The explanation for this obviously is that there's a very high rate of reversion. So you can think of this as a bank of switches. In the first generation, they are all in the on position. Then randomly, you toggle some to the off position. In the next generation, again, you randomly toggle those. But of course, you will toggle some of those back into the on position again. And this has important implications for Darwinian selection because reversions will reduce the increase in epimutation distance over time. And if it's very high, selection essentially cannot work. Selection cannot change the frequency of these epimutations. Very similar picture for differentially methylated regions shown here, so not just individual positions, but entire regions. These are quite rare, so rather than 30,000, only 30 per strain. But again, we see many of those in more than one strain. Okay, so last couple of slides. Okay, last couple of slides. This was all done in the lab. What you really want to know what's going on in nature. Now, these experiments take quite some time, but we were fortunate. We discovered an experiment that's about 200 years old, and that was started when white settlers introduced Arabidopsis thaliana to North America. About half of Arabidopsis thaliana in North America is genetically mostly identical. So we sequenced the genome of 20 of those strains. They only have a total of 1,600 mutations. To the root are about 200 mutations. I told you 1 mutation per generation per genome, so about 200 years since they diversified. And the relatedness fits well with the geography. But we did this so that we'd be able to look at the methylome. And what you see here is that lines that are related to each other genetically, they're also highly related to each other epigenetically. So there's nothing really magical about epigenetics. And these are now plants that have lived in the wild in quite stressful and changeable environments. So to summarize the first part of my talk, told you about the trade-off between defense and growth. And one conclusion there is that having too many disease-resistant genes can be dangerous. You're well protected against pathogen, but at some point you just start to kill yourself, which isn't very good either. Then the second part, I told you about mutations and epimutations. There's a similar frequency of DNA mutations and differentially methylated regions, but frequent reversions of epialleles will likely interfere with Darwinian selection. And our conclusion, or hypothesis, if you will, is that epigenetics probably is mostly important in short-term adaptation, but probably does not play much of a role in long-term evolution. All right, my last slide. I want to put in a short plug for a new venture being started by Howard Hughes Medical Institute, by Wellcome Trust and the Max Planck Society, a new online journal, eLife, headed by, this must be the competition trying to trying to take the word away from us. Won't work, you guys. Headed by Randy Scheckman, who was editor-in-chief of PNAS before. We have some very good ideas how we're going to change publishing, especially how we're going to change reviewing, going to really streamline that so that you're not strung around by editors for a year or 2 years, only to learn in the end that perhaps your work will not be published at all. If you want to learn more about this, if you have some very high-class work you are looking for an outlet to publish it in, please come see me after the presentation. Thank you, and thank you, Max Planck.
Okay. Any question before the break? I have a question. You said that too many disease-resistant genes can be dangerous. Can be applied to any other family gene within plants? I mean, resistant genes is easy because you face with a pathogen. But what happened with, I don't know, transporters or any other kinds of family genes?
So this is specifically these immune receptors. So the immune receptors also evolve in a special way because they're arranged in tandem clusters. They often have transposons in these clusters. They internally repeated. So there is a lot of diversity that's being generated at immune receptor genes in plants all the time. Much of that diversity is probably not yet functional, if you will. Plants just explore the potential phenotypic space to be prepared for whatever new pathogen they will encounter. And in principle, the more you have of those, the better you are prepared for new pathogens. But to many, you might recognize plant proteins, and this is what happens in these cases that we are investigating.
Okay, we have time for one more question, okay? You have to fight. Yeah.
I can repeat it if you shout, Regina. How do these epi alleles actually originate? So what's the process where an entire region changes at once? Because I told you both about changes at single positions, and I talked about these regions. So I would say so far this is not really not quite clear, but actually my colleague David Balcom has a very nice story where one of these differentially methylated regions, they discovered a mechanism how this switching between two different states might occur. And it's still unpublished, so I can't tell you, but hopefully we'll soon read about it.
Okay, I think we have to stop. And we can continue just discussing during the coffee break. And I think, I'm not quite sure, but I think we'll be back at 5:30, okay? We'll have the last two talks after the coffee break. Okay, thank you.